Hopefully you got your morning coffee, which may be hormetic to keep you at least a little bit alive for my talk, which might get boring. Uh, uh, it's an incredible meeting, honestly. As a postdoc or as a student, I'm really honored to share my data with you. But the interaction in general was amazing, and I mean it sincerely. Thank you, Ed. Like, I've been to some aging meetings and big meetings. It's really easy to get lost in all those big wigs and everything, but I never got the chance to interact with scientists and students the way I did here. So it kind of reminds me of the Gordon conferences as well. So, and it has been extremely rewarding in terms of the knowledge I kind of, accrued or gained just yesterday. I'm hoping to look forward to getting more of the insights today. So I'd like to share two stories with you. So one came from David Sinclair's lab where I'm working, and then one in collaboration with Jay Mitchell, who is in Harvard School of Public Health. It's kind of intersect at angiogenesis and health aspect of it. I'll try to do my best to give you overview of that. I don't think I have time to go into the details. And we're trying to communicate two papers on this work right now. So this work started at Lenny Gurente's lab, who's a pioneer in sirtuin field at MIT, and then I moved to Harvard in David's lab to finish the work. But the catch was then I had to move to Australia here, University of New South Wales, to finish some of the work. Of course, he told me that it will be really good for you because you can have a healthy lifestyle and it can extend your health span. There's really cool stuff to do in Australia, but he never talked about the lifespan that can shorten with all these poisonous and venomous things in Australia. But I mean, there's another person here from Australia, and he can he can support me on that. That it has been an incredible experience. They have a really good scientific community. They are really good research support. I don't want to teach the old dogs new tricks here because it has been redundant since all the talks yesterday. That all you are trying to do using hormetics is to extend healthy lifestyle or health span, right? And if you go to the aging meeting, one of the things that changed over time was initially started talking about lifespan. Cynthia Keenan talked about, oh, you make DAF2 mutants, you will extend the lifespan by 50%. If you go to her talks now, or at least last October, you'll see she's changing her focus and she's talking more about health span. And there's really nice work done by Heidi Tissenbaum, UMass Worcester, where she kind of tracks down each worms during their lifetime, and she showed how they are living, how their movement is, how healthy they are when they are dying. So, and you can all appreciate the fact that when we get old, we want to be that beetle, not the first one. And if you want to drop dead, we want to drop dead just like that, okay? That's it. And there's no, I mean, the problem here is although the humans are living longer compared to the 100 years ago because of vaccination, less infection, our lifestyle, but still, the last 10 years of our life are still miserable. We suffer through so many diseases. Our lifestyle are really sedentary. We cannot do exercise and all these things. The key is actually to push that boundary. You can get a lifespan extension, but push that healthy boundary so that even the last few years are actually, mobile for us. And that's the focus of my talk is today. Here again, you know that there's a frailty is one of the problem with aging, and it's associated with a lot of diseases. These are only few of them, and a lot of them actually happen due to vascular dysfunction. Even some of the neurodegenerative diseases starts with some vascular dysfunction in the body. There's some paper on that, and it's surprising that still if a person with peripheral arterial disease or cardiovascular diseases go to a clinic, the doctor will still prescribe him exercise. But what happens if you have a 90-year-old dad can actually exercise? What happens to patients like that who has a hip surgery and can't exercise to cure the peripheral arterial disease kind of thing? So that kind of made us to think about, can there be an exercise mimetic drug that can mimic the effect of exercise in these patients who can't actually voluntarily exercise? And also we know that the body's response to exercise goes downhill with aging, and that's a major concern, like vascular remodeling especially. So there have been a lot of focus on, there's modulators of anti-aging kind of thing. That's a word I don't want to use, but more like exercise mimetics. Like diet is one of them, and there's a lot of focus by Ronald Evans on AMPK activators, PPAR delta activators, and David has been working on resveratrol for a while, But there's no clear-cut answer, like like what can be used for exercise mimetic yet. So talking about the diet, and there's a classical example of the diet that extends lifespan from worms to non-human primates. It's known. That's a Wisconsin study and NIH study on the rhesus monkeys. This calorie restriction is if you restrict the calorie intake, it's known to extend lifespan. And there's different players in there, and still a lot of controversy going on, which is more predominant than the others. So I don't want to go there. But the problem here is if you ask somebody that, okay, go under calorie restriction when you're 25, they'll get bitter. They'll get bitter before it gets better. And it's very, very hard to push this agenda there. I mean, if you think about it practically and pragmatically, it's not possible to ask somebody to go under calorie restriction. So working with Jim Mitchell's lab, we started focusing on methionine restricted diets. So there have been some studies done in 1993 who showed that even if you don't decrease the calorie intake, but if you take away methionine from your diet, there's a massive lifespan extension in rats and also in the mice as well. And just 2 years ago, J. Mitchell showed that this is mediated through CGL, a protein that actually produces H2S under stress in the body. And using, in this paper, they showed that methionine-restricted diet can actually protect the mice from pre-perfusion injury in liver through CGL and H2S. So we got interested in finding out what happens with methionine-restricted diet and angiogenesis and health in general, because there's a lot of connection between H2S and angiogenesis and endothelial cells. This is one part of the story, and another part of the story where we intersect is we're also trying to look at how SIRT1 can be modulated in the same pathway to look into how it can help in angiogenesis and health. So, so what we did, basically, we put the mice under 2 months of MR, and we found there's a massive increase in VEGF in the muscle and slightly in liver as well. And what it does, it basically increases, it's hard to see. It actually increases capillary density in the gastroc muscle here. And you can block that with an inhibitor which blocks VEGF receptor, and you can see by blocking it, there's this increase, MR-mediated increase in vasculature in the muscle is kind of like gone right now here. It shows that it's actually VEGF-mediated. And this kind of diet can also be beneficial in ischemic conditions, like you can see here, the classic hind limb ischemia model, where there were preconditions for 1 month, and then the femoral ligation surgery was done, and the mice actually recover really well under MR diet. So when we took CGL knockout mice, we see that this MR benefit is vanishing there. So it clearly indicates that it's CGL mediated. And then this is another interesting aspect of it. So these mice were the wild types of the CGL knockout were actually put under mild exercise training. And you can see here, and we all know that vascular remodeling with exercise is a very predominant thing. And you can see that when the CGL is not there, there's no vascular remodeling happening in the muscle, which clearly shows that CGL is a big player in terms of exercise and also even in the sedentary conditions in terms of vasculature in the muscle. So what's happening actually is there's two aspects of it. There's a muscle aspect of it, and there's endothelial cells that are interacting with the muscle. And what we find that there's an increase in H2S signaling in the endothelial cells with MR diet. Okay? That made us to think that, oh, something is happening in the endothelial cells in response to MR, or in terms of MR plus exercise that's increasing the capillary density or number of blood vessels in the muscle. So we looked into it, and we found that endothelial cells are actually a big producer of hydrogen sulfide gas. And you can see here, this is a lead acetate assay done. This PAG is a CGL inhibitor, and the inhibitor, there's no increase in H2S, whereas if you stimulate the cells with VEGF or methionine-restricted media, there's an increase in lead acetate. The more black means more H2S. And this is a classic speedoid assay done with the endothelial cells under control condition or methanorestricted media. You can see there's an increase in sprouting in these cells, whereas when they are knocked out of CGL, there's no increase in sprouting, showing it's totally kind of blocking the angiogenesis there. And if you just treat the cells with sodium hydrogen sulfide, there's an increase in migration in these cells, showing that it's somehow mediating, the migration of the cells and also the sprouting and angiogenesis of the cells. And I'm not going into the details, but you have done a very detailed mechanistic analysis of this, and then we have found that the main role H2S is doing here is actually blocking mitochondrial complexes and increasing glycolysis. And that's how it's promoting. And glycolysis is very important for endothelial cells when they actually sprout or migrate. So, and then we found that the classic mechanism here is actually it's the amino acid starvation response, which activates GCN2, and it's hypoxia independent. That's the cool thing about it. So this pathway basically activates CGL, which releases H2S, and that blocks mitochondrial electron transport chain, increases glycolysis, and promotes angiogenesis. So that's the story, and then that's the story of the MR. And then what came out from the story was we also found that SIRT1 is somehow required for this angiogenesis, which is H2S independent in MR conditions. And we kind of got more interested into it, and we're trying to find out, like, okay, what's happening with SIRT1, because it's known from various work by Stephanie Dimler, Michael Potente, Carmeliet, that SIRT1 plays a very big role in angiogenesis. So to address that, what we did, we took two different models. One where SIRT1 is knocked out, specifically in the endothelial cells. They don't have SIRT1 in the endothelial cells. And the second one is where SIRT1 is overexpressed in the endothelial cells. These are classic Cre-Lox model. So that the knockout model is ESCO, and the overexpressing model is ESTO. And you can see by 6 months of age, the knockout actually have less capillaries in their muscles. Whereas if you overexpress, there's a increase in vascular bed in their gastroc. This is quad, but then I have analyzed quad and gastroc. In all of the muscles, the vascular bed increases with overexpression. So that's good, but then we were trying to find out what it does in terms of physiology. That's what we were more interested in. So what I did, I actually subjected these mice to exhaustive endurance. Okay, I tried to find out how long they can run. Okay. So this is the results, and we found that just like how the results look like for capillary density, it actually mimics their exercise capacity. So the knockout mice actually run less, whereas the overexpressing mice actually run longer. So in a way, the knocking out SIRT1 just in endothelial cells mimicking early aging. Whereas the overexpress, you're actually delaying aging kind of, or age-related symptoms here. And just a point to note here, I also analyzed, every other aspects that can affect exercise capacity that you can think about, like, muscle mitochondria, fiber type. That's usually come to our mind, right, when you think about somebody running better. And I did a thorough analysis of it. I did not see any change in, mitochondrial number, mitochondrial protein amount, fiber types, or even when you take the isolated fibers, you can measure the oxidative capacity. I did not see any changes in them, showing that it's mainly predominantly happening because there's a change in vascular bed in the muscle, which is kind of interesting. So the question is, okay, so how do we translate? Because on aging labs, so how do you translate it to aging? Those were genetic models. Can we modulate SIRT1 pharmacologically? And the good thing about SIRT1 is it can be. So there's smaller activators that we know that speakers talked about. There's resveratrol, which is one, but then we also found that one of the fuel for SIRT1 is NAD. It's an NAD-dependent deacylase. And so the NAD actually goes down with aging. It decreases from 12 months onwards. So if we measure so this paper in Anna Gomes from our lab showed that from 12 months onwards, it steadily declines. It becomes less than 50% compared to the control by 20 months of age. And she also showed that the NAD, SIRT1 can be activated if you replace NAD using a precursor called NMN. So the question was okay, can we do the same thing? Can we reproduce our genetic model in an old mice using the pharmacological agent like that. So what we did, we actually treated really, not really old, but like 18 months, 20 months old mice with low dose of NMN for 2 months. And we found that there was a massive increase in number of capillaries in the muscle, in the quadriceps and also in gastrocs, just when they were only for 2 months of treatment. And you can see what it does. It basically increases the microvasculature in the gastroc. You're looking at the gastroc here. This is a mouse leg. This is a contrast ultrasound where you inject microbubbles through the tail vein, and you can see increase in signal going through. If there are more vascular bed, there were more signal coming in, and you can actually measure peak enhancement, which is a measure of relative blood volume, and you can see there's an increase in blood volume there. And this is another technique where we looked at oxyhemoglobin contents. So, and muscle is really hypoxic. You can see it's only like 60% oxygenation there, but it increases by 15%. It actually is huge if you think about how hypoxic it is. So there's an increase in oxygenation of hemoglobin, and there's also increase in the microcapillaries. And the key thing about this mechanism is it's actually SIRT1 dependent. So if you take wild-type mice, you see an increase with the same treatment here, where if you knock out SIRT1, this is actually whole body even, okay? This is even better. If you knock out SIRT1 from the whole body, there's no increase in microvasculature, showing that the NMN effect is clearly SIRT1 dependent. What are the other things it can do? This is just aging model, sedentary conditions, give them NMN, it's increasing vascular bed. So this increase in vascular bed or capillary density can actually make them run longer as well. So I subjected these mice to 2 different tests. It's a low intensity exhaustive endurance, high intensity exhaustive endurance. And in both of these tests, the NMN treated mice actually performed way better than the vehicle treated mice. So it kind of recaptures exactly how our genetic model sits. And again, there were no significant changes in terms of muscle mitochondria, fiber types, or even oxidative capacity of isolated fibers. So in addition to that, we also put the mice through femoral ligation experiments. And you can see here, we actually took wild-type and knockout mice. And this was, the capillarity was measured using peak enhancement, using contrast ultrasound that I showed you in the last slide. And then it's a massive increase with NMN treatment after femoral ligation after like 20 days. But when you knock out SIRT1, there's no increase, showing that this is primarily through SIRT1 again. And you can see there's an increase in capillarity in the muscle where whenever you knock out SIRT1, there's not much increase with NMN. So another experiment, we actually subjected these young mice to exercise and also treated them with NMN. We initially found that at a relatively young age, like at 6 months, there's not much effect of NMN there. It's a very small amount. And we were trying to push the boundary. I'm like, okay, what's stopping it? Like if you give NMN, and this is what hormesis come into effect, because what dose you should give to push the boundary, So it's actually, can you combine 2 hormetics together to have a synergy there? And this is a classic example, like how this works. So exercise, if you think is at hormetics, it will push the boundary of capillary to some extent. But can we combine that with NMN, another hormetics, and push it even further? And that's exactly what's happening here. The sedentary, they have a very similar capillarity, where when you exercise them, it goes up. But when you combine them with NMN, it actually goes even farther. And in the experiment, what I did, I actually treated them with axitinib just to show that NMN or SIRT1 is working downstream of VEGF. So again, when the mice were treated with axitinib in diet, which is a VEGF receptor inhibitor, you don't see that increase in capillarity with exercise. It's showing that, so you have the MR on the top, there's VEGF coming, and the SIRT1 is down. So you're actually modulating 2 different pathways, upstream and downstream, with 2 different agents, which is pretty cool. And so again, we did a thorough analysis of what's going on, and we did a lot of assays in vitro and in vivo, ex vivo. We found that this is primarily due to the fact that NMN is promoting angiogenesis to SIRT1. So this is the classic sprouting assay where you take aggregates of cell and put them in collagen matrix, and they actually like form sprouts. So when the study was done, we kind of got into in collaboration with J. Mitchell, and he was working on sodium and sulfide, and we asked, like, can we even push it further? Can we actually take NMN and NHS together? Can it have a beneficial effect? And we had no idea whether it would work or not. So we took really, really old mice, 32-month-old mice, like almost equivalent to like 90-year-old human being. And we treated them with small dose of NMN and really, really small dose of sodium hydrogen sulfide and through drinking water. And what happened was it actually massively increased their capillarity in their muscle, like almost doubled it, like 100% increase. And when I put those mice under exhaustive endurance, just think about you're asking your dad to basically run with Usain Bolt, okay, something like that. What it did was it actually almost doubled their exhaustive capacity, exercise capacity as well. So we have some mechanistic detail about that. What we found that is actually a balance between angiogenesis and apoptosis, and it shows actually blocks apoptosis to SIRT1 pathway. So there's actually there's not only a lot of capillaries in this dual treatment, with this dual treatment, but there's also a decrease in apoptotic endothelial cells in the muscle or in the muscles as well with this kind of dual treatment. So I'm not going into the details in the mechanism because of the sake of time, but we have some knowledge of the fact that this is mediated to Notch1 inactivation. So SIRT1 is actually inactivating Notch. That's why it's promoting angiogenesis, and there's a lot of connection with Notch intracellular domain and angiogenesis. So what happens here is when muscle, which is a rich source of VEGF, releases VEGF under normal sedentary conditions or under stress conditions like ischemia or exercise, the signal gets picked up by the endothelial cells, and if SIRT1 is sitting there, it kind of acts as an amplifier. It amplifies the signal. But the problem there is it can act as an amplifier as long as it has the fuel or the petrol to run. But then petrol here is NAD, which is the co-substrate. But the problem is it runs out with age and with time. In some way, pharmacologically, you can actually put back that fuel, refill the gas tank, it can still start working. And there's not only one hormetic, but there can be another hormetic like H2S that can actually add on the top of that, and that can have an additive effect as well. So, and that can actually cause, this is just one aspect of healthy aging, which is neovascularization in muscle, but it can have effects in other body parts and other tissues as well. So I think that will need more detailed analysis as well. But the key here is it's not only one hormetics, it's actually multiple hormetics combined. And from this meeting, it's clear that there's a synergy between different hormetics, and we have to harness that energy. So it's synergistic hormetics. Can we just, I know it gets incredibly complex at this point, but we have to agree that there cannot be one hormetics that can give us a beneficial effect of healthy lifestyle. There has to be combination of one or two. And this is a time where people have to appreciate that, we have to play with the combinations. Dose not all biphasic dose for not only one, but two or three, even more. And that's where maybe we can go, or we can get the end results of getting a really, really good healthy lifestyles or lifespan as well. So I'll just stop there. I don't know, I think Ed can comment on that, whether it's a good term to use or not. Or I just wanted to throw out in there just to get your opinion, because you're experts in hormetics field. I just want to get your criticism, get your idea, like what do you think about that? Because it's kind of, I kind of feel passionate about it. I believe in hormetics. I've been working on it for long, and I think there's not one, but there has to be like more than one hormetics that can actually help us live longer, live healthier. I'll just end there. I just want to show a quick slide where I work, and not here, of course, I don't work here. No, on the top of this is the Sydney Harbour Bridge. You can go on the top of it, of the bridge, take a tour. It's a really cool place. You can see a Sydney Opera House, and that's Sydney there, and UNSW is somewhere here. But I'm incredibly lucky to go and work there and finish my work. But throughout this process, doing this work, I've been incredibly lucky to work with different people from different institutes and have a different collaborations ranging from MIT to Harvard to UPenn. And also, it's good to have money when you're doing research and running like cohorts of, I think I ran like 300 mice or more. I don't know how much money I wasted in Lenny's lab, but it has to be more than 300 mice. So yes, funding is very important. So and this was done because of funding from NIH in US and NHMRC from Australia. Thank you very much. So this paper is in, there's two papers under revision right now. just to let you know that I'll be going into job market this fall. So um, if you have any jobs opening up in university, please keep me in mind. I'll be happy to send my resume. Hopefully this paper makes through. I'm extremely gratified to present my work here. I look forward to your comments and questions. Thank you.
Questions? I'll start, hand it over to you.
Sure.
Two quick questions. Obviously, with a methionine-restricted diet, that is a very, very low protein diet.
Yes.
So you're also taking a look at muscle integrity, and of course this speaks also to our next speaker.
Right, yep.
So if in fact it's a severely methionine-restricted diet, you also then run the risk of inducing borderline kwashiorkor malnutrition. And you also see muscle wasting because of proteotropic muscle wasting. How much of a methionine-restricted diet is advocated for humans, for example, to induce this type of effect?
I think it was 40% less or something less, similar to the calorie-restricted diet. So you're not talking about, we're not taking away methionine restrict like totally. It's like, I think we're reducing it by 40% at least.
And with 40% methionine restriction with regard to protein-based methionine products, which
Correct.
are seen as
Exactly.
Okay.
And I think if you take it totally, it's actually bad because
Sure.
because the essential amino acids.
Well, particularly in the ages, because then you
Yeah.
see muscle wasting. And
Exactly.
as a consequence, then you're also going to see
I agree.
problems with bone.
I agree. So the rat or the mice under MR, which live longer, are actually smaller. It may be because of sarcopenic kind of effect, but the living longer, that can be a different story. But you are right. So initially when the studies were done, they were actually like really, I think more than 60% methane restriction. It was severe.
Yeah.
But now we're reducing it down again, dose response.
Because the translational implications are huge, particularly for an aging population, that you also, as you do with so many things, you run that
I agree.
critical balance.
Yeah.
At what point do you then begin to induce muscle wasting and protein malnutrition? And although you may be seeing angiogenesis and compensatory effect, the actual sort of cellulogenic aspect of angiogenesis that is counterbalanced by...
I agree. Yeah, it may be like lowering of oxidative fibers, that's why it's compensating with vascular
Correct.
capillary bed. Yeah, I agree with that.
No, interesting stuff.
Yeah.
Good. Sir.
So, very interesting that H2S mitochondrial interaction.
Yeah.
And yesterday we heard that maybe that it can actually be an electron receptor. So, you showed that it blocks electron transport through oxygen phosphorylation. So, is it still maintaining the health of the mitochondrion during that time, or is it basically suffocated the mitochondrion?
So the question is, so here I showed that H2 is blocking mitochondria, whereas H2 is known to be electron donor. So during the treatment, is it still maintaining mitochondrial integrity? We haven't done that analysis yet on the muscle fibers. I think it needs to be done. I think that's one of the reviewer asked the question as well. But you have to understand the key here is there's like a subtlety of two different models. H2S, that's mediating all these effects, endogenous, coming from CGL inside the cell. And we're trying to reproduce that kind of effect in vitro using exogenous H2S. So it's very different. The H2S, if you do in immediate, the half-life is only 5 minutes. So transiently what we see that it just blocks it. Hormetic response, at ATP gets low, AMPK gets activated. AMPK gets activated only for 5 minutes. The mitochondria shuts down. That kind of effect. I don't know whether sustained H2S will actually, that's happening with the endogenous H2S, it has the same effect or not. So you're part of the mechanisms of endogenesis. It may not be all the mechanisms of endogenesis.
Sure.
Sorry, I can
query and comment about studies you are doing. It's not only on your study, but on others that you are trying to show that these molecules are promoting against that migration. And these are exactly the targets of anti-cancer, cancer receptors, gate these two pathways. So you are promoting the pathways which are actually cancerous.
Yeah, that's like, the question is like, we are promoting angiogenesis. What happens in cancer? Because that's one of the big targets for cancer, and that's the question we get all the time. So we look into different cancer models, like again model in liver. We do not see abrogation of cancer. Cancer do not go up surprisingly, and the NMN effect or of the MR effect kind of primarily only limited to the muscle only. I think it's because of cell-to-cell communication. So MR actually has beneficial effects on cancer. MR actually reduces on cancer. And some of the H2S drugs are known to
studying